Okay, hello everyone. I'm Jing Zhang from Dr. Jacob's group, and my research is mainly about developing new statistical methodologies on the area of computational biology, and this work is jointly in Dr. Jacob's lab and Dr. Liang Jin's lab in computational and molecular biology. And first, when we look at the figure, what do you think about? It is not the spectrum of the audio, and it is not the system random error. Actually, it is a genetic change for diabetes. Some people just identified that there are 6 changes, 6 mutations in our genome that will cause the diabetes. And then under this line, we know that from the DNA sequence, some of them are selected and translated to the premature mRNA sequence, and these translated part will be responsible for the protein codings. But amazingly, we found that some of these silent regions, which is not coding for the protein sequences, has been responsible for some severe genetic disorders. So my job is to identify which part of these silent regions will be functional, and this is not only a fundamental scientific problem, it is also a billion-dollar price problem in the drug industries. So my work has been in 3 layers. In the first layer, we'll see what are the regulation mechanisms from the DNA sequence to the mRNA sequence. In the second layer, we developed 2 methodologies, one called GP-seq and another is GP-EM, to measure how much DNA has been generated from mRNA. In the third layer, we have another model to analyze how much mRNA has been generated into proteins. In this way, we have a whole pipeline understanding how the DNA sequence has been generated into proteins. Okay, in the first problem, we have the sequence of the DNA of DNA in our genome, and we use the partition functions and to predict the topology of the structures of the mRNA sequence, and we can also have some energy functions to predict the stability of such structures. And we found that the structures has severe affections on the selecting of which part of the DNA sequence has been translated to proteins. And in the second project, among the billions of nucleotides in the genome, we are searching for very, very short functional elements in the silent regions. And we develop some complicated statistical methodology named varying coefficient regressions to solve this problem, and it works excellently on human genome. And another project, how to quantify how much DNA has been translated to mRNA. Here is how we count the number of translations. And we just utilize the generalized Poisson distribution to quantify this problem, and we adopt EM algorithm in the following analysis. And our method can improve the result up to 80%. And then one of the ongoing projects is to set up some models to understand how much mRNA sequence has been generated into proteins. Okay, this is mainly my work. Thank you, and any questions?